All right, so what I wanted to talk about is the portal and some other updates that I have. Oh, sorry. Yeah, that makes sense. So when we started talking, or when we started working on SATB2, we created the website SATB2gene.com. And what we had originally envisioned was more of a resource for everybody kind of thing, for researchers, families, and whatnot. We have photos here, Claire and others that are here with us, and you can tell that those photos were taken several years ago. But anyway, so the main issue with the website, or the few issues that we have had in the past, is that first is that there's a fair amount of overlap. If you Google SATB2-Associated Syndrome, either one of the websites come up. And then we have had some overlap, right? So the resources, do you go to this and that website for like letters or brochures or whatever you want to give to your provider? So there's a fair amount of overlap. And now with each foundation having its own website, even more so. So like Australia has its own website, its own resources, and France and Italy and whatnot. So there's a fair amount of overlap. And then the other part of it is that when we started it, we wanted to give back all the information that we were collecting, but it's all static, right? So if you go to the website and you want to query, there's some files that you can download, like, okay, what's the frequency of these and what are the mutations that you have? But it's very static, so you can download a file and do whatever you want with it, but it doesn't allow you to interactively kind of thing. And the same if you want to do your own research, and we've been talking about different mutations and whatnot, if you want to compare it with others and whatnot, it's not been a good way to do that until now. So introducing the portal, which actually I got the final URL today. Let's see if I can do this correctly. Okay. So this at least as of right now is the website that we'll be using, at least for now. I foresee that the SATB2gene.com website in the end is going to finish and just route whoever is searching to this portal. So what you have here, I'm just going to quickly walk you through it. So you have the main tabs here. You have basic information. You have the sort of information for families. Then you're going to have a tab for genetic changes specifically. If you want to compare or what sort of main symptoms are related to what genetic change, this is the genotype, phenotype. And then we have some information about the registry and the clinic. I'm just going to quickly walk you through some of these. So under basic information, you have on the left side sort of the history of the gene and kind of how we came about it. Each hyperlink here will take you directly to the paper that is referring. And then here in the middle, so this information now is compiling not just the registry, but I keep tabs on cases that are reported in the literature, and I try to use the same kind of template of how I summarize that information so you can see where that information is coming from. So not to overwhelm the website with a lot of data, so we kind of selected a few things to show, basically. So this is not just registry data. Again, this is registry plus literature data. So you have, under population breakdown, you have your gender, age. If you click under speech, you have some of the things that we're talking about as far as when they start talking, when they start walking, so different things, and it will tell you the number of individuals that we're gathering that data from, because we don't have the data for everybody, so it will tell you, it's not 310 for everybody that we have all the information, so it will just tell you the main ones, so the same with cleft palate or whatnot. So that is the basic information part of it. If you go under families, so we have, again, just some basic information, kind of a summary. We have videos. This is from the foundation, and this is from the 2019 meeting. a talk that I did, very broad, just kind of summary of information. Here it will have, again, just kind of the main features. Now, if you look here at the top, and this is, again, as I was saying, with the different foundations, and I have talked to, as each foundation is introduced, I have given presentations to each one of them in the past, and I have spoken to their leaders because it's so sub-branched that it's a little bit harder for somebody looking for resources if they're routed to 10 different websites. So that I've been bringing up to attention, because it'll be nice if it's just one combined website that has all the resources that apply to everybody. And then if you live in Italy, then you click on Italy, and then you get the resources that are specific for you, kind of thing. So if you go here on the family organizations, when I started working on the portal, these are the foundations that were up and running and had a direct link to it. So we have here the US one, Australia, and the Europe side. So these are hyperlink, and Spain. So these are hyperlinks. So if you click on any of these, it will take you specifically to each foundation's website. And then on the resources are the PDF documents that you're all familiar with, letters to providers and whatnot. So next, if you go under variant analysis, then this is one of the new things. So I'm going to actually test. I was just talking to some of you about how to interpret each variant and whatnot. So if you have your genetic testing results, again, if you remember, there's a C dot and a P dot that it will tell you kind of where the mutation is, assuming that it's not a deletion, again, which is when you're missing the whole gene. So if you go, let's say 715, C to a T, the idea is that you do either one, so either the C dot or the P dot. If you notice here, I did the C dot, it's already telling me that it's going to be okay, I'm going to click search here. Hopefully this works. So yeah, so if I click search here, it's telling me the type of mutation it is. So this is what we call a nonsense mutation. That's what the stop gain means. And then here at the bottom, now it's going to tell you, okay, so for you guys, if you were into, not just you, but researchers, clinicians, if you're like, okay, let me see if this specific change has been reported before, this is how you will do it. So you look at the variant, and then it will tell you, okay, well, this variant, which is a 239, it has an asterisk, sometimes an X, sometimes a TER is the same. So now you can compare apples to apples here with some of the information as far as, okay, well, when did they start talking? When did they start walking? This is all de-identified, so you wouldn't be able to tell who's who here. Did they have seizures and whatnot? And then if you keep scrolling here to the bottom, so you can download an Excel file. I'm just going to try it here and see how it happens. Which is going to be the master file here for the specific thing that you're looking for. So now you can kind of look here if you want it. And I know the foundation research part of it was asking me what data is available. Is it available for everybody? So this is going to be able to be downloaded by whoever, you guys or researchers, clinicians, and whatnot. So that part is cool that way. So, and then if you go, let's see, if you keep going here, and then you go under genotype, phenotype, now you can, so that previous part, the variant analysis, that's if you know the specific change. Now, this genotype, phenotype, now you're looking at, okay, let me just compare. That was, for example, that specific change that we were looking at, that was what we call a nonsense mutation. Okay, so now I'm not interested in just 239, which is where that nonsense was, but I'm interested in all nonsense. So I will just, for example, here, filter it by nonsense. And now we're comparing all the nonsense. It will tell you how many there are. And then if you look here, this is the same graphic we've been seeing throughout. So Dr. Fish had similar slide showing like the length of the protein, which this is, again, the same kind of thought process that I was trying to have yesterday and explain to you guys. So it starts here at number 0, and it goes 733 building blocks long, so it will tell you the position of where each individual variant is. So we're looking here again because I'm filtering by nonsense. So these are all the nonsense variants and where they are located. If I hover over any of these points, it will tell me how many times that variant has been seen. That 239 is one of the most common ones, so so it says 18. So 18 people have had exactly the same change. So that is just for the nonsense variant. So I'm gonna try a different type of variant just for the fun of it. We're gonna try missense, because this goes back to what I was talking about yesterday. So if I'm filtering by missense, okay. So now you have, and yesterday we were talking about where the majority of the missense mutations are located. So now if you filter by missense, here at the bottom, it will very nicely have like the density of where the variants are located. So you see in that green area here, which is one of the main domains that binds to the DNA. We were talking about this yesterday. So you see the vast majority are located here, which is why the peak is so downward. But you see they're located in those areas mainly that bind the DNA. So this one's over here, and then this one at the end. The problem when we get genetic testing results, and this has happened a few times, is how to interpret, and I have told families this before. So when the variant or when the change in the gene is located elsewhere, and there's just one patient, and it's in the middle with no specific area that we know has a function, then what do I tell that family? So that's the whole point of why we're trying to understand how this works. So, all right, and then if you go under phenotype interface here, then the same. I'm filtering here by missense, so this is going to tell me where the variants are located. And then this is all for all the missense variants now. It's telling me how many words they spoke, what the breakdown is, who had seizures, abnormal behavior, teeth, or whatnot. So likewise, if I was to say, hey, let me see who had a cleft palate, for example. So I can do that and filter by cleft palate, for example. So these are who had a cleft palate in the database, and it tells me who had it. It's going to, all these genetic changes had a cleft palate, for example. And again, I can download the Excel file here at the bottom, and the common link here, again, is that they all had a cleft palate. So if you wanted to filter some more, then that's how you will do it. Brain MRI, the same. You get the gist of what I'm trying to say. Okay, and then the last part of it, so the registry, there's just some basic information here. If you click on participate, it will just directly take you to your email default program, and I think it's Katie's email that is going to be listed as the default, who the email is going to be directed to. And then in the about is just how the data came about and who contributed. This is done in collaboration with a really broad group of people from Cleveland Clinic. You see Katie and myself here, and Jenny Lee, who many of you know, was instrumental also in providing feedback on how to display the data, and she's been very helpful. Okay, so that's the portal. So with that, I'm gonna go back to my presentation. Okay. We already went through this. This was in case I couldn't do the live demo. Okay, so other projects and updates. So as far as other projects, so Dr. Sanchez and I were hoping to get some 3D photos. That is going to be on hold. We had some technical difficulties, so we're going to stay tuned for that. Dr. Fish was talking briefly about this earlier. So we are interested in differences in the development of the jaw. And it's a combined effort of looking at her mice, the fruit fly, which she mentioned, the group in Baylor, and then a lot of photos that we have over time. So we are combining all that data to try to understand how the jaw forms differently in SAS, basically. She also alluded to this, so I'm going to repeat it. So the group in Baylor is looking at how each change has a different role and how it affects the SATB2 gene. I put the asterisk here because it's a project founded by the foundation. So that is looking very promising from what I hear. And by the way, all these projects are projects that I know about or that I'm directly or indirectly involved. So there's probably many more that I'm just not aware. Okay, so concurrently, I've been working with a group in the Netherlands. Dr. Fisher is his name, his group. So there's different ways, and we've been talking about this, and some of you have asked me, okay, and actually I have my list of unanswered questions from previous talks that I was going to try to go through. So one of you asked, okay, how do we know if a mutation is gain of function or loss of function and whatnot? And that is what all this is all about, honestly. So you would think that if you have what I was saying, we call a null mutation. So those are nonsense frame shifts, splice sites. If you have those type of mutations, you would think that because the protein is cut short, then that is a loss of function. In theory, that's what should happen. But we know that that might not be necessarily the case because the protein is still being produced. So what we don't know is that little bit of protein, instead of having the 733, if you have a much shorter protein, is that sufficient to do something? Or is that worse because it's going to now mess up the good one? Because you have two copies. You have one good one and one that is deficient. So that's the whole point of what we are trying to understand. For missense variants, as I was explaining yesterday, so you have the normal length of the protein. It's just one building block that is misspelled, but it's in an area that is critical for the function of the gene. So those, do those behave as loss of function, gain of function? That's the part that we're trying to understand. And there's different ways to do it. So I'm collaborating with Dr. Fisher's group, and I gave him a list of some of the variants that we know, especially missense variants are the ones that we're going to try to figure out. A list of variants that we have seen multiple times that I have a lot of data on, like patient data, so I can correlate it. Okay, this is more severe or less severe. So I'm working with them on that, not using mice, not using fruit fly, but rather using cells and how it changes the very small structure of where the SATB2 gene is, what is happening kind of thing. Concurrently, whenever you get genetic testing results, the lab that does it, the computer automatically tells you what is predicted it will happen with a given change. So we're trying to figure out, is there a common way to figure out what kind of consequences a change could have? What is the best computer program to predict that? So that we're working on as well. Dr. Fish alluded earlier about her stem cell studies. So the part that, I guess what she was trying to say is that from the stem cells, she was trying to get bone cells. And then now she's kind of changing gears towards brain cells, neurons. So that's one part that she's going to be working on. So she mentioned also that we have shared the cells with 4 different groups. So one of the groups that we have shared the cells with is Dr. Chen, who's in California. She's actually doing something really cool, which is the crazy thing about stem cells is that you can basically manipulate them in a way that you can make them be whatever you want them to be. So it can be bone cells, it can be neurons, brain cells that is, or whatever you want. So she's going to try to work on the neuron part and almost like create a small, what we call an organoid. So that means a smaller brain in a petri dish created from stem cells. So that's what Dr. Chen is going to be working on, which is critical to understand what happens, because most of the research that has been done with SATB2 is either mice or it can be human, but it's not the same because it was a mutation that was created by CRISPR, which is what Dr. Fish was saying, as opposed to you're born with this mutation. So it's a bit different. So she's going to be working on that. That, by the way, is going to take, I don't know, a few years, I will guess. We, Children's Hospital, Arkansas, we're working on different ways to measure bone density. Some of you that have been to clinic with us are aware of this study. So we're looking at bone markers in blood, and we're trying to find a different type of X-ray that perhaps is more sensitive than the bone density. It's called a quantitative CT. The problem that we're running into is that you do need to stay fairly still for that, and of course, it's nearly impossible with the kids. So we're working on it. So we've got some data. We're hoping to get more. It has an asterisk too, because it was also founded by the foundation. We briefly talked about this yesterday. So we're looking at our data with bone density. We were trying to understand, okay, why the US population has not as often low bone density. So we're looking at that. I'm probably going to start measuring phosphorus routinely, just for the things that we talked about yesterday. We already are doing alkaline phosphatase and all the other stuff. So, and there's another marker, and this is from the same two patients that I was referring yesterday that had the abnormal phosphorus. There's another marker that we use that is called FGF23 that has been elevated in those two kids that had the low phosphorus, which could indicate a completely different process of how or why the bone is abnormal. So that will require a separate source of funding, the FGF23. It's not like I can order it in clinic, but the phosphorus will be pretty easy to do. Okay, and then yesterday also we talked about the metabolism study, which Dr. Fish had a slide on it too. So as I briefly explained yesterday, we are doing the same kind of study, but now on the skin cells, to document that those abnormalities can be repeated not just in blood, but in other cells. I know that you guys were asking, actually, I forgot, sorry, your name. You were asking about doing the metabolism study in brain cells. So if we generate neurons from the stem cells, this will be how we will do it. Use the same platform that we use in blood and the same platform that we use in skin. We will use it in neurons if we had them. So that's been the key. So if we replicated it now in 3 different cell lines, then we will know it's much more legit than just the one sample. So for the skin part of it, that's also a foundation-sponsored project. So we're working on that.